All right, so now in Unit 3, we're going to move on to glycogen metabolism, which is covered in Chapter 16. And so we've split this into two lectures. The first lecture here, we're going to talk about glycogen structure and synthesis and breakdown, and then the regulation of glycogen metabolism. The second lecture, we're going to focus on mainly clinical pearls, glycogen storage diseases, because those are definitely a hot topic for biochem exams and board exams. So first, just to start with an overview, a lot of these concepts with glycogen we've really talked about in previous lectures, especially when we talked about glycolysis. But again, this is just really to further the story of carbohydrate metabolism and metabolism in general, and really focus in on how glycogen really fits into that story. So glycogen, as you know, is the stored form of glucose. The primary function of glycogen breakdown in the liver, remember it depends on which organ we're talking about, is to provide glucose for other tissues. So the liver does not use glycogen during fasting. It actually uses fatty acids, as we've talked about. So it breaks down glycogen for glucose for tissues such as the brain. Muscle glycogen, however, is the opposite. It's broken down during exercise and fasting to provide additional glucose for glycolysis so that you can have ATP production to power muscle contraction. So the structure of glycogen, this is kind of just this is a cool zoomed out view of what it looks like. It just gives you the idea of how branched and complex the structure of glycogen really is. So it's a branched polymer of glucose, as we say here. And these residues are linked together by an alpha-1,4 glucosidic bonds for the most part. So we'll come down here. Here's a glucose molecule. And as you can see, here's a bond right here. So remember, glucose has 4, 5, 6 carbons. So here's the 1 here. And then if we go here, 2, 3, 4. So 1, 4. So alpha-1,4 glucosidic bond. So that's this bond right here. Now, on average, every 10 residues or so, there's a branched form by a different type of bond, this alpha-1,6 glucosidic bond, and that's shown in this diagram here. Now, this diagram is just simplistic because it just wants to show you the different types of bonds. In glycogen, this would usually be more spaced out. So again, 1, 2, 3, 4, 5, 6 here, and then this is the first carbon here. So 1,6 alpha-1,6 glucosidic bond. Now, glycogenin is a protein that's core to glycogen structure. This is a lower yield topic, but we just kept it in here to really just give you the complete picture of how glycogen is synthesized and how the structure is formed. So glycogen is bound to this protein called glycogenin, and you can see that here with this cool 3D rendering of the protein structure of glycogenin. And it serves as really a primer for growing glycogen changes. So glycogenin catalyzes the addition of 8 alpha-1,4 glucose units, like that bond we talked about on the previous slide, the alpha-1,4 glucosidic bond, to itself. So it catalyzes the addition of these 8 units to itself using uridine diphosphate or UDP glucose as a glucose donor. And so it donates glucose from 8 of these to form these 8 units. Then glycogen synthase adds glucose molecules beyond these 8 initial molecules that were added to glycogenin. And this is how you form glycogen. However, glycogen synthase is only active when it's bound to glycogenin. And really beyond that, that's all you really need to know for this protein. All right, so for glycogen synthesis, we're going to start with glucose in the bloodstream. And remember, it gets taken up into the cell. GLUT2 in the liver, GLUT4 in the muscle, enters into the cell. And once it enters the cell, remember, there's that investment of ATP to phosphorylate glucose to glucose 6-phosphate. This is carried out, remember, by glucokinase in the liver and then hexokinase in most other cells. Now, and remember, glucose 6-phosphate is a very key intermediate. It can go to a number of different directions, including glycolysis. If it's not going to go towards glycolysis and go towards glycogen synthesis, what happens first is glucose 6-phosphate is converted into glucose 1-phosphate by an enzyme called phosphoglucomutase. And this is a reversible enzyme. And then glucose 1-phosphate is converted into UDP glucose by combining it with UTP, uridine triphosphate. And this is catalyzed by the enzyme UDP glucose phosphorylase. And this forms UDP glucose, which, as we talked about previously, is a glucose donor. This is an irreversible reaction as well. And so UDP glucose phosphorylase is an irreversible enzyme. So then the glucose from this UDP glucose is added to a growing glycogen chain and forms a new alpha-1,4 linkage that we talked about. This is catalyzed by the enzyme glycogen synthase. Now, this transfer of glucose from UDP glucose to a growing glycogen chain, this will only occur at a pre-existing glycogen primer of at least 4 glycosyl units, which are found on glycogenin, which is why that protein is so important. You can't do this until you have that primer essentially attached to that protein glycogen. All right, so you're synthesizing a chain of glycogen. As you notice in the structure of glycogen, it's a highly branched structure, and that's advantageous, and we'll talk about why in a second here. But to form that, you need an enzyme called branching enzyme. So branching of glycogen occurs at alpha-1,6 linkages, catalyzed by branching enzyme, not to be confused with debranching enzyme, which is an enzyme we'll talk about in glycogen breakdown, two different enzymes. So you'll notice here we have a diagram here, a very simple cartoon, where we have a chain of glycogen here, and then you have a branch here. These branches occur at these via 1,6 linkages. So these right here are 1,4 linkages to form this chain, and as well here, these are 1,4 linkages as well. And then at the branching point, these are alpha-1,6 linkages here. And so these branches are formed by taking the terminal segment of a glycogen unit, 7 glycosyl units. So you'll see we numbered it out here, 1, 2, 3, 4, all the way to 14. And so you have to take from a branch here. You can't just take from the growing chain. You take from one of the branches that are already formed. So you take these terminal 7 units, as you can see here, 7 through 14, and you'll take this unit and then add it to the C6 position on a glucose molecule. Could be on the same chain as we're going to show here on this diagram. Could be another chain as well. So you've moved it to here, and you've created a new 1,6 linkage here. So now you can continue to grow this branch, you can continue to grow this branch, and then you can continue to grow the primary chain as well. And so by allowing for branching enzyme to carry out this function, you allow for more rapid rate of synthesis, and then by forming this highly complex branch structure of glycogen, you allow for a more rapid rate of degradation of glycogen as well, which is really important when you want to break it down to give yourself glucose to metabolize it for energy, which is important in the fasting state and then also during exercise as well in the muscles. All right, so now that you've synthesized glycogen, the next step is to break it down. And fortunately, you don't need to use any ATP to break down glycogen, which is a process called glycogenolysis. And so that's nice when, this is usually in a fasting state and you want to break down glycogen to glucose as rapidly as possible. So it's nice that you don't have to use up any ATP around to do that. The main player here is glycogen phosphorylase, and this catalyzes the breakdown of glycogen at that alpha-1,4 linkage, so that linkage that holds them together. So that's, here and here. So the 1,4 linkages here. And this generates glucose 1-phosphate. So you have glycogen phosphorylase that approaches at these branches here, and it starts out at the terminal end of the branch, and so it kind of just works its way down. So it'll cleave, generate a glucose 1-phosphate, cleave again, and glucose 1-phosphate, and it'll do that all the way until it gets to 4 glucosal units from the branch point. So here's the branch point here. We're 4 units out, as you can see, and at that point, glycogen phosphorylase has to stop. What happens is that 3 of those 4 remaining units are transferred to the chain that the branch stems from. And so this is carried out by an enzyme called debranching enzyme, as we indicate here. So you can see that here. Here's those 3 final glucosal units transferred to the primary chain here. And then this remaining glucosal unit is actually cleaved to free glucose. And so this can just go right into glycolysis if you're in the muscle. If you're in the liver, it can then just be exported out to be used by peripheral tissues. And one thing to note here, as we say, is 10% of the glucose in glycogen is directly converted to this free glucose. So this is about 10%. And then the remaining 90% is first converted to glucose 1-phosphate. So that glucose 1-phosphate, once you've generated it, it's rapidly converted to glucose 6-phosphate, which is a key intermediate. Now, what happens next depends on what tissue you're in. If you're in the muscle, it can then go to glycolysis, because it's typically, you're going to be breaking this down during exercise or prolonged fasting, and that muscle needs to use that glucose for ATP production. Versus if you're in the liver, there's an enzyme called glucose 6-phosphatase, which essentially cleaves off this phosphate group at the 6th position to give you free glucose. And then this free glucose can exit the cell and enter into the bloodstream to be used by other tissues. Now, it's important to note that the muscle does not express glucose 6-phosphatase. A couple of reasons for that. Number one, remember the muscle during exercise is not like the liver. It uses the glycogen it breaks down. It's not breaking down glycogen to glucose and then exporting it to other tissues like the liver is. So that's one reason where there's no point to have to be able to convert it back to glucose, because it's going to use it right there. The other reason is that this is a key enzyme in gluconeogenesis. It's actually the very last step of gluconeogenesis. And again, muscles don't carry out gluconeogenesis. They don't synthesize glucose, as we've explained in depth in the glycolysis lecture. It uses that glycogen directly into glycolysis to generate ATP again. So again, there's no point for the muscle to have glucose 6-phosphatase. All right, so now that we've talked about the structure, the synthesis, and the breakdown of glycogen, now we want to talk about how this all fits into the greater picture of metabolism and the story we've been telling here. And so that where that fits in is the regulation. And given the importance of glycogen, it has many levels of regulation. That includes influences from hormones, energy metabolism, glucose levels, muscle contraction, and caffeine. And so before we get into the different states, such as the fed state, the fasting state, or how exercise affects these different scenarios, we want to just remind you of some key concepts here that really help you, again, to further understand how glycogen fits into this whole story and how it impacts other metabolic processes. So during fasting, blood glucose is low, and that induces the liver to break glycogen down to provide glucose for peripheral tissues that primarily rely on it, such as the brain. The other key concept here, number 2, is in the fed state, insulin is elevated, so you have high insulin, and that ensures the uptake of glucose into the cell. And then what that's going to do is stimulate the formation of glycogen, because again, as far as the body's concerned, it doesn't know when the next meal is coming, and so when you have this abundance of glucose around, you want to store it for periods of fasting and starvation even. And then 3 here, during exercise, muscles are rapidly breaking down glucose via glycolysis, and so this creates an increased need for glucose, and so that's going to induce glycogen breakdown as well, so that can be used in the muscle to provide ATP for muscular contraction. So first, the fasting state. So a key thing we've talked about throughout these lectures so far is the fasting versus the fed state and how that impacts regulation and how these processes are being carried out. So on each of these slides, we're going to have the fasting state, the fed state, exercise. What you want to look at is the end goal here. What is the end goal? So if you're fasting, you need glucose. So you're going to be stimulating the breakdown of glycogen. On the flip side, you don't need to be storing more glycogen because you're in the fasting state, and so you're going to be inhibiting glycogen synthesis. So this is really important to remember these two end goals, because then all of this really falls into place and makes sense. So you're trying to increase breakdown, inhibit synthesis of glycogen. So during the fasting state, as you know by now, glucagon is increased. Glucagon comes down here and stimulates adenylate cyclase to convert ATP into cyclic AMP. So you have high levels of cyclic AMP. And then, as you know, cyclic AMP stimulates protein kinase A, which has 2 main effects here, as we'll talk about in a second. And so again, remember the end goal here. You want to inhibit glycogen synthesis. So let's first look over here. Protein kinase A phosphorylates glycogen synthetase using a phosphate from ATP, and that renders glycogen synthetase inactive. So then you have essentially blocked glycogen synthesis here. Let's come over here. So protein kinase A takes a phosphate from ATP and phosphorylates an enzyme known as glycogen phosphorylase kinase. So this is a separate enzyme from glycogen phosphorylase. So it phosphorylates glycogen phosphorylase kinase. That makes it active. So then glycogen phosphorylase kinase then phosphorylates, it's in the name, glycogen phosphorylase, which then makes glycogen phosphorylase active. And then glycogen phosphorylase can carry out the process of breaking down glycogen to glucose 1-phosphate, which can then be used in the cell for glycolysis. Or in the state of the liver, it can be converted into free glucose, which can be exported for use by other tissues. Now, in the fed state, it's the opposite. You've just had a meal, you have plenty of metabolic fuel around. So in this period, you want to actually block glycogen breakdown. And that makes sense. You have plenty of metabolic fuel around. You don't need to be immediately breaking down glycogen for free glucose. On the flip side, as far as the body's concerned, it doesn't know when the next meal is coming. So if it has an overabundance of glucose, it's going to want to store it for periods of fasting. So on the flip side, it's going to be stimulating glycogen synthesis. So again, it's really important. Remember these 2 end goals. The rest of this will fall into place. So during the fed state, you have increased insulin, as you know. And insulin's going to impact a few different things. So first, insulin's going to block adenylate cyclase. So you have decreased levels of cyclic AMP, decreased activation of protein kinase A. And then on the flip side, it's going to activate an enzyme called protein phosphatase 1. or PP1, which as you can see here, has effects on all three of these major regulation points here. And so as far as PP1 is concerned, let's go back to our end goals here. So you want to be stimulating glycogen synthesis because you have an overabundance of metabolic fuel. So you look at protein phosphatase 1 here, it's going to take off this phosphate group from glycogen synthase and make it active again. And so that's going to stimulate synthesis of glycogen from free glucose around. On the flip side here, PP1 is going to take this phosphate off of glycogen phosphorylase kinase, which will render this inactive, which will then decrease activation of glycogen phosphorylase. And then any remaining active glycogen phosphorylase will have that phosphate taken off by PP1, rendering it inactive. And then as a result of that, you will be blocking glycogen breakdown. So again, it all comes back to these two end goals to help service the fed state. All right, so during exercise, you're using up a lot of glucose, producing as much ATP as possible. So you have depleted levels of glucose eventually as you continue to exercise. And so you're going to need to break down glycogen to help give you new glucose to break down in glycolysis. So again, your end goals are you want to stimulate glycogen breakdown. And since you're breaking glycogen down, you don't want to be synthesizing it. So you want to inhibit glycogen synthesis because again, you don't have some overabundance of glucose around. You're using up as much as possible to help power exercise. And so again, you want to stimulate glycogen breakdown, inhibit glycogen synthesis. So during exercise, you have increased epinephrine, increased adrenaline, that makes sense. And epinephrine stimulates adenylate cyclase, which is then, as you've talked about, is going to stimulate the synthesis of cyclic AMP, which is then going to activate protein kinase A, which we've talked about this before. So briefly again, protein kinase A is going to phosphorylate glycogen synthetase and inactivate it, thus inactivating glycogen synthesis. That's in line with our end goal. And then over here, protein kinase A is going to phosphorylate and activate glycogen phosphorylase kinase, which is then going to go on and activate glycogen phosphorylase by phosphorylating it, which is then going to activate glycogen breakdown. And the thing we'll point out here is epinephrine has the same exact effect on these enzymes as in the liver as it does here in the muscle. And because the same thing in the liver doesn't use the glucose that's broken down from glycogen. It actually ships it out to peripheral tissues. And then during exercise, it's probably going to be skeletal muscle because you want to be providing even an extra little bit of support for powering those muscles. Another interesting, unique thing in the muscle is muscle contraction increases the intracellular level of calcium. And so you have increased levels of calcium here. And so calcium does a couple things. First, it activates phosphofructokinase 1 or PFK1, which is a very key regulatory step in glycolysis, as we talked about. So it's going to increase glycolysis. The other thing is it binds to the calmodulin subunit of glycogen phosphorylase kinase, which activates it. So it's going to come over here and activate glycogen phosphorylase kinase, which is then going to further activate glycogen phosphorylase, which is going to further activate glycogen breakdown. And so again, this all stems back to the end goal here, where you want to break down glycogen to provide more glucose for glycolysis to power that exercising muscle, while in turn inhibiting glycogen synthesis. So the effects of glucose itself, first we'll look here at glucose 6-phosphate. Now, if glucose 6-phosphate is a negative allosteric regulator of glycogen phosphorylase, so it's going to come over here and inhibit it. And then it stimulates glycogen synthetase. And it makes sense. If you have elevated levels of glucose, you want to inhibit glycogen breakdown. So the end goal here is you want to stimulate glycogen synthesis while inhibiting glycogen breakdown. And that makes sense. If you have an overabundance of glucose, you certainly don't need to break down any more glycogen to provide you glucose. And here, same thing, glucose itself, elevated levels of glucose inhibit glycogen phosphorylase, which inhibit breakdown in the liver specifically. The one thing that we'll point out though is as the cell reaches its full glycogen capacity, because remember there's a maximum point, glycogen will inhibit conversion of glycogen synthetase to its active form, so it'll inhibit continued synthesis. Because there's only a maximum glycogen storage capacity, if you will. And then remember in the liver, once you've reached that, then you switch over to storing your abundance of glucose as fatty acids in adipose tissue. So as far as the cellular energy level, what effects that has? So first, let's look at ATP. So that would be a high energy state. And so ATP is a negative allosteric regulator of glycogen phosphorylase. So it's going to come down here. And that makes sense. If you have high energy levels, you don't need to be breaking down glycogen to provide more glucose for glycolysis. And so what it's going to do is favor glycogen synthesis. On the flip side, if you're in a low energy state, you're going to have higher levels of AMP, and AMP is actually a positive allosteric regulator of glycogen phosphorylase. So that's going to favor glycogen breakdown because it's a low energy state. You're going to break down glycogen to provide glucose to help produce more ATP for energy. And then lastly here, we'll just mention that caffeine plays a small role here as well. And this can actually show up on tests, both biochem exams, board exams. So it's definitely something that should not be ignored. And so caffeine, as we show here, inhibits the enzyme phosphodiesterase. Phosphodiesterase breaks cyclic AMP down into AMP. So by inhibiting phosphodiesterase, you maintain that level of cyclic AMP at an elevated state for longer. So you increase cyclic AMP, still maintain increased activation of protein kinase A here and here. And if you remember, protein kinase A inhibits glycogen synthetase, which inhibits glycogen synthesis. On the flip side, it activates glycogen phosphorylase kinase, which then activates glycogen phosphorylase, which activates glycogen breakdown, as we say here. So end goal, caffeine helps stimulate glycogen breakdown. All right, so that closes out our first lecture on glycogen, where we talked about the structure, the metabolism, the regulation of the metabolism. In the next lecture, we're going to talk about glycogen storage diseases, which is a very high-yield topic.